Today we are diving deep into one of the most incredible structures in all of biology. We're talking about the plasma membrane. It's this super thin, super active, and, surprisingly intelligent barrier that literally defines the very edge of life itself.
So let's just start with a huge question right off the bat. What actually separates the complex, organized world of a living cell from, well, everything else? Where exactly is that line?
And here is it. This is our answer, the plasma membrane. But look, it's not just some simple wall. It's what we call a selectively permeable barrier. That's a fancy way of saying it's an active gatekeeper. It is constantly, meticulously deciding what gets in, what has to stay out, and what gets shipped away. And that control, that's the foundation of everything for a cell.
Okay, so if it's a gatekeeper, how is it built? To really get how it works, we've got to look at its structure. And the best way to think about it is something called the fluid mosaic model. We're going to break down exactly what that name means, piece by piece.
It all kicks off with this special little molecule, the phospholipid. And its secret? It's something called amphopathic. It's a great word, and it just means the molecule has a kind of split personality. One part, the head, absolutely loves water. But the other part, the tails, can't stand it. They're water-fearing.
And this picture shows you exactly what happens because of that split personality. You drop these phospholipids in water, and they automatically arrange themselves into this beautiful structure, a bilayer. The water-loving heads point out, facing the water that's both inside and outside the cell. Meanwhile, the water-fearing tails all tuck into the middle, away from the water, creating this stable, oily core.
Now here's the really wild part. This barrier isn't rigid. It's not a solid wall at all. The source material gives us this perfect analogy. A membrane is about as fluid as salad oil. It's in constant motion. Which brings us right back to that name, the fluid mosaic model. So it's fluid because all those phospholipids and proteins are constantly shifting around sideways, kind of like people in a crowded room. And it's a mosaic because you have this rich patchwork of all different kinds of proteins studded throughout that sea of lipids.
And when I say constant motion, I mean it. The speed is just mind-boggling. Get this, a single phospholipid can move sideways about 10 million times per second. Compare that to the incredibly rare event of it flip-flopping all the way to the other side of the membrane. That might only happen once a month. This sideways flow is absolutely essential.
So you might be wondering, how does the cell keep that salad oil from getting too runny when it's hot or freezing solid when it's cold? Well, in animal cells, the hero is cholesterol. It works like a fluidity buffer. When things get worn, cholesterol actually holds the phospholipids back a bit, keeping them from getting too liquidy. And when it gets cold, it gets in the way, preventing them from packing together too tightly. It's the perfect temperature manager.
All right, so the membrane is this fluid, temperature-controlled structure. We've got that down. Now let's get to the main event. Let's see how that structure allows it to do its most important job, controlling traffic. It's time to see the gatekeeper in action.
So, broadly speaking, there are two main ways things get across the membrane. Passive transport costs the cell zero energy. It's like rolling downhill, from a high concentration to a low one. Active transport, on the other hand, is the workout. The cell has to spend energy, usually a molecule called ATP, to push things uphill against their natural flow. The most basic kind of passive transport is just diffusion. You've seen this before. Imagine dropping food coloring in a glass of water. The molecules just naturally spread out until they're evenly mixed. Small things like oxygen and carbon dioxide can do this right through the membrane. No help needed.
But what about stuff that's too big or has an electrical charge? They can't just squeeze through that oily middle layer. For them, the cell uses something called facilitated diffusion. It's still passive, still going downhill with the flow, but it uses special proteins that act like selective tunnels or shuttles. A perfect example is something called an aquaporn, a channel just for water that lets it zip through way faster than it could on its own.
Okay, but what happens when the cell needs to bring something in that's already rare outside, or it needs to pump out waste that's already crowded out there? It can't just rely on things floating in. It has to fight against that natural downhill flow. It has to spend some energy.
And here is the superstar of active transport, the sodium-potassium pump. It's constantly at work, so let's walk through it. First, 3 sodium ions from inside the cell latch onto the pump.
Then an ATP molecule comes along and gives the pump a little packet of energy. That energy forces the pump to change its shape, and poof, it spits the sodium ions out of the cell. Now, with its new shape, 2 potassium ions from outside can bind. That causes the energy packet to pop off, and the pump snaps back to its original shape, releasing the potassium inside the cell. Now here's the crucial part. For every single cycle, the cell spends 1 ATP to pump 3 positive charges out and only bring 2 positive charges in. This tiny imbalance, happening millions of times, builds up an actual electrical voltage across the membrane. And that voltage is vital for things like sending nerve signals.
Controlling ions is a huge deal, but what about controlling the most important molecule of them all, water? This is where we have to talk about osmosis, and it is absolutely make or break for a cell's survival.
To really get osmosis, you need this one concept, tonicity. It sounds complicated, but it's really just a way to compare the concentration of a solution to the concentration inside a cell. It tells you whether that solution will make a cell gain or lose water. You've got 3 scenarios: isotonic, where it's balanced; hypertonic, where the solution is more concentrated; and hypotonic, where it's less concentrated. In this slide just shows you the consequences perfectly. I mean, look at that animal cell. You put it in a hypertonic, salty solution, water rushes out and it shrivels up. But put it in a hypotonic solution, like pure water, and water rushes in so fast that it literally bursts. Now look at the plant cell. Its tough cell wall completely changes the game. In that same hypotonic solution that made the animal cell explode, the plant cell just gets nice and firm, turgid. And that's exactly what a plant needs to stand up straight. It's just a perfect example of how structure dictates function.
So, we've gone way down into the microscopic mechanics, but let's zoom back out. Why does any of this matter in the grand scheme of things? How does this connect to our world?
Well, for one, those proteins we saw in the mosaic, they're not just doors and tunnels. They are the ultimate multitaskers. They can be enzymes, they can receive signals from the outside world, they act like little ID tags so cells can recognize each other. They do so much. They're really the workhorses of the membrane.
And understanding these proteins isn't just academic, it's medically critical. Take HIV, for instance. To infect one of our cells, the virus absolutely has to latch on to a specific type of protein on the cell surface. There are actually a small number of people who are naturally immune because their cells are missing that exact protein, so the virus just can't get in. That knowledge is the entire basis for developing drugs that block that exact same interaction.
Okay, let's wrap this all up. What have we learned? The cell membrane isn't just a baggie holding the cell's guts in. It is a fluid, active, intelligent barrier, a fluid mosaic. It uses both passive, no-energy transport, and active, energy-burning transport to control every single thing that comes and goes. And in the end, that power to choose, that selective permeability, is the very thing that draws the line between the random world outside and the incredible, organized miracle of life inside.
So I'll leave you with this final thought to chew on. This fluid, constantly shifting barrier is what defines every single living cell on the planet. So what does it really mean, both biologically and maybe even philosophically, that life itself chooses to exist right on such a dynamic, ever-changing, and permeable edge? Pretty amazing stuff. Thanks for watching.